Zinc, an essential trace mineral with a profound impact on human health, plays a crucial and often underappreciated role in cellular maintenance and regeneration through its involvement in autophagy. While commonly known for its contributions to immune function, wound healing, and DNA synthesis, zinc is deeply integrated into the machinery that governs autophagosome formation and lysosomal function, two critical stages of the autophagic process that are responsible for clearing cellular debris, misfolded proteins, and damaged organelles. Through these roles, zinc helps to maintain cellular homeostasis and protects against the metabolic and oxidative stresses that accumulate with age and chronic disease. Autophagy, at its core, is the cell's self-renewal program, and it relies on a tightly orchestrated sequence of events: the identification of cellular waste, the encapsulation of that material into autophagosomes, and the eventual fusion of these vesicles with lysosomes where degradation and recycling take place. Zinc contributes meaningfully at both ends of this sequence. First, it supports the expression and function of key autophagy-related genes involved in the early formation of autophagosomes. Without sufficient zinc, this initial step can be impaired, leading to delayed or insufficient clearance of cellular byproducts. In studies involving zinc-deficient cells, researchers have observed reduced autophagic activity, increased oxidative damage, and the buildup of dysfunctional proteins, all hallmarks of accelerated cellular aging and decline. Equally important is zinc's role in lysosomal acidification. The lysosome is the terminal organelle in the autophagy pathway, where engulfed materials are broken down by highly specialized enzymes that require an acidic environment to function. Zinc assists in the regulation of proton pumps and other ion transport systems that maintain this acidic pH within lysosomes. If lysosomal acidification is disrupted, whether by aging, metabolic imbalance, or nutrient deficiency, autophagic flux stalls and waste products can begin to accumulate within cells. This phenomenon is linked to a range of chronic conditions, including neurodegenerative diseases like Alzheimer's and Parkinson's, where defective protein clearance plays a central role in disease progression. Zinc also acts as a signaling molecule and stabilizer in stress response pathways that are closely tied to autophagy. For example, under conditions of oxidative or inflammatory stress, intracellular zinc levels can rise transiently, activating transcription factors that upregulate genes involved in autophagy and antioxidant defense. This adaptive response helps cells survive and recover from damage. Furthermore, zinc has been shown to modulate the activity of the mechanistic target of rapamycin, mTOR, the nutrient-sensing pathway that negatively regulates autophagy. In zinc-deficient conditions, mTOR activity may remain inappropriately elevated, thereby suppressing the autophagic response and reducing the cell's ability to clean itself. From a neuroprotective perspective, zinc's support of lysosomal function and autophagic efficiency is especially important. The brain is particularly vulnerable to autophagic dysfunction due to its high metabolic demands and the long lifespan of its neurons. In healthy brain tissue, zinc helps facilitate the turnover of synaptic proteins and the clearance of aggregated tau and amyloid beta peptides, processes essential for cognitive function and memory retention. Several studies have highlighted that disturbances in zinc homeostasis correlate with impaired autophagy and increased neuronal vulnerability, suggesting that optimal zinc intake may help support long-term brain health through the preservation of autophagic integrity. Dietary sources of zinc are diverse and accessible, though the bioavailability of zinc varies significantly depending on the food matrix. Shellfish, particularly oysters, are among the richest natural sources of bioavailable zinc, followed by red meat, poultry, and fish. Plant-based sources such as legumes, chickpeas, lentils, beans, seeds, pumpkin, sesame, sunflower, whole grains, and nuts also provide zinc, though they contain phytates that can inhibit absorption. Soaking, sprouting, or fermenting these foods can improve zinc bioavailability. For individuals with higher demands, such as those with gastrointestinal issues, chronic stress, or plant-based diets, supplementation with zinc gluconate, citrate, or picolinate can ensure adequate intake. As with many micronutrients involved in autophagy, balance is key. Both deficiency and excess of zinc can disrupt cellular function. While mild to moderate supplementation can be beneficial for those with insufficient intake, Long-term high-dose zinc, especially above 40 milligrams per day, can interfere with copper absorption and should be approached cautiously and under professional guidance. When combined with other autophagy supportive nutrients such as magnesium, vitamin D, and polyphenol like resveratrol or EGSG, zinc forms a synergistic part of a broader nutritional strategy for cellular resilience. It also complements lifestyle interventions like fasting and exercise, which place increased demand on autophagic systems and benefit from the enzymatic support that zinc provides. In conclusion, zinc is a vital enabler of autophagy through its support of autophagosome formation, lysosomal acidification, and stress-responsive gene expression. These actions ensure that cells can properly identify, sequester, and degrade damaged materials, maintaining optimal function even under conditions of metabolic or environmental stress. With its wide range of dietary sources and its essential role in so many cellular processes, zinc stands as one of the most important micronutrients for preserving autophagic efficiency, supporting immune defense, and promoting long-term metabolic and neurological health.